Good morning, everyone. Today we are going to see about the histology of male reproductive system. So in this class, we are going to see about the structures of male reproductive system. In gross anatomy itself, we have discussed the gross features of organs in the male reproductive system. Just we are going to refresh the things now. Then we are going to see about the microscopic features of organs in male reproductive system. So these structures only we are going to discuss today. They are testis, epididymis, vas deferens, prostate, seminal vesicle. So these are the things we are going to see now. First, before going to the microscopic features, we will see about what are the structures in the male reproductive system. So this is the sagittal section of male pelvis. It shows the urinary bladder here. Here you can see the prostate gland. Here you can see the penis, scrotum. So within the scrotum, you can see the testis. This is the male gonad, ovoid-shaped structure. It lies in the scrotum. Then you can see the comma-shaped structure, that is the epididymis. Epididymis continues as vas deferens here. You can see the long tube-like structure. This vas deferens joins with the seminal vesicle and it forms the ejaculatory duct. This ejaculated duct opens into the prostatic part of the urethra. So this is the prostate gland. Okay, so in this class, we are going to see about the testis, epididymis, vas deferens, seminal vesicle, and prostate gland. Okay. Now we'll see about the testis. You know, testis is the male gonad. So it lies in the scrotum, outside the abdominal cavity. You can see the ovoid-shaped structure. It has the upper pole, lower pole, anterior border, posterior border, medial surface, and lateral surface. Posterolateral, you can see the epididymis. So here you can see the vas deferens. This is the cord, spermatic cord with the contents. Okay, so it lies in the scrotum. What it produces? It produces the sperms, and also it secretes the testosterone hormone. It's a male reproductive hormone. When you see the testis, it is covered by 3 layers. They are called as capsules of testis. You can see here this blue color, no, that is the visceral layer of tunica vaginalis. This is parietal layer, visceral layer of tunica vaginalis. So in between that cavity will be there. It contains the fluids. Then the next layer is the tunica albuginea. This is the thick white fibrous tissue. It is adherent to the testis. When you cut open, within that you can see the tunica vasculosa. So what are the coverings of the testis? They are the tunica vaginalis, tunica albuginea, and tunica vasculosa. Now we'll see the cut section of the testis. The things we already discussed in gross features. So I already know, just I want to refresh the things now. You can see this is the ovoid-shaped structure. Okay, so this is anterior border, posterior border. You can see this is the thick white fibrous tissue, that is the tunica albuginea. So this tunica albuginea projects inside the testis near the posterior border and it forms the mediastinum testis. Okay, from the mediastinum testis, you can see numerous fibrous septa extending. This fibrous septa, it divides the testis into numerous compartments. These compartments, they are named as lobules. You can see here separate compartments. No, these are called as lobules of testis. Okay, lobules of testis. So within each lobule, what you are seeing here, you can see some coiled tubules. No, they are nothing but the seminiferous tubules. Seminiferous tubules. Okay, these are the structures. These produce the sperms. Okay, when you see the seminiferous tubules, they have a coiled part and a straight part. Okay, so these are the straight tubules. So sperm produced from the seminiferous tubules, they drained through the straight tubules and they enter into this sieve-like structure. This is called as the retitestis. So here they will be like a branching and they are joining again. So this part is called as retitestis. From the retitestis, you can see efferent ductules, 12 to 20 efferent ductules will be there. These efferent ductules, they join and form the head of the epidermis. From here it continues as body. Then here you can see the tail of the epidermis. This tail of the epidermis, it continues as the vas deferens. Okay, you understand this now? Now we can see again. So testis is covered by the tunica albuginea. Okay, this tunica albuginea projects in the posterior border of the testis and it forms here the septum that is called as mediastinum testis. From the mediastinum testis, numerous fibrous septa is extending and it divides the testis into lobules. Around 200 to 300 lobules are there. Each lobule is lined by tunica vasculosa. Okay, each lobules, they will be lined by tunica vasculosa. Within that, you can see 2 to 3 seminiferous tubules. You can see 2 to 3 seminiferous tubules. These seminiferous tubules, when it's near the retitestis, you can see some straight tubules. So these straight tubules, they join and form the retitestis. From the retitestis, you can see 12 to 20 efferent ductules. These efferent ductules, they join and form the epidermis. Epidermis continues as the vas deferens. Okay, you understand this now? Now, in this picture, you can see this is the testis. You can see the coverings, parietal and visceral layers of tunica vaginalis. In between that, you can see the cavity. Okay, then this blue color structure is the tunica albuginea. This tunica albuginea, what happens? It projects in the posterior border that is called as retitestis. From the fibrous septa arise. This divide this into numerous compartments. These are called as lobules. Within the lobules, you can see the seminiferous tubules. Now, we have to discuss the histology part. No? So, we have to make a section and we have to see through the microscope. So, when we make a section in this part, okay, so you can see seminiferous tubules. It's a tube-like structures. When we cut a tube, you can see the round ends, no? So like that, here it is convoluted, highly convoluted, no? So you can see in one section, numerous cut sections of seminiferous tubules you can see here, like this. Okay, in this picture also you can see, this is the outer parietal layer, visceral layer of tunica vaginalis. Then you can see tunica albuginea. Then tunica vasculosa will be there. So this is one lobule contain numerous seminiferous tubules. Okay, these are the seminiferous tubules. So from here only sperms are produced. So we are going to see about the seminiferous tubules. Then in between seminiferous tubules, you can see some interstitial tissue that also we are going to see. So in testis, after the covering, what you have to read about the seminiferous tubules and interstitial tissue. Okay, so when we make a section, you can see like this. So this thick white fibrous tissue, that is the tunica albuginea. The next layer, this vascular layer, is the tunica vasculosa. You can see the septum, fibrous septum, it's also carrying the vessels. So this is one lobule. Okay. So this is the lobule. Within the lobule, what you can see? Numerous cut sections of seminiferous tubules, because it's a coiled tube. When we cut within one section, we can see the numerous cut sections of seminiferous tubules. Okay, these are the seminiferous tubules. So here only sperms are produced. You can see here also, see sperms are produced here. No? So we are going to see about the seminiferous tubules individually, and we are going to see about this interstitial tissue. In between seminiferous tubules, some tissues are there. No? That also we are going to study now. So first we'll see about the seminiferous tubule. So seminiferous tubule, it will be around 70 to 80 centimeter in length, 150 micrometer in diameter. So the diameter will be 150 micrometer, and it is covered by a fibrous tissue, fibrous connective tissue. So it will be covered by a fibrous connective tissue. So when you see inside, it is lined by two types of cells. One is named as spermatogenic cells. Another one is the Sertoli cells. Sertoli cells, they are also called as supporting cells, because these Sertoli cells, it's standing like a pillar. In between the pillars, you can see numerous spermatogenic cells. These are the spermatogenic cells. Okay, so when you see the seminiferous tubules, within that you can see spermatogonium and also Sertoli cells. The tall cell is the Sertoli cell. These numerous cells, they are in multiple layers of that is the spermatogenic cells. Okay, so this one only we are going to see now. First, we'll see about the spermatogenic cells. Spermatogenic cells, they are starting from below. So this is a part of seminiferous tubules. You just see this first layer. These are the spermatogonium. So these are the germ cells. So it is there. From the spermatogenic cells, spermatogonium, due to mitotic divisions, the primary cells, spermatocytes, they are produced. Primary spermatocytes. From the primary spermatocyte, secondary spermatocytes are produced. From the secondary spermatocytes, spermatids are produced. From the spermatids, spermatozoa is produced. Okay, so what are the spermatogenic cells? Spermatogonium, primary spermatocytes, secondary spermatocytes, spermatid, spermatozoa. These are the spermatogenic cells. Usually in sections, the secondary spermatocytes you can't see because this half-life is very less. Okay, so when the primary spermatocytes divides into secondary spermatocytes, the secondary spermatocytes, it becomes spermatid soon. So in sections, the secondary spermatocyte will be difficult to identify. Mostly you can see spermatogonium, primary spermatocytes, then spermatids you can see, then spermatozoa. Okay. So they are arranged from basal lamina to the lumen. So from the basal lamina to the lumen, they are arranged one by one. So first spermatogonia, then spermatocytes. Spermatocytes, they are two. They are primary spermatocytes, secondary spermatocytes. Then you can see spermatids, then spermatozoa. Okay. So these are the spermatogenic cells. Okay. So we'll see. This is a section of seminiferous tubule. We are going to read about the seminiferous tubules now. So from basal lamina to the lumen, these are arranged. So just see this group alone because these are the spermatogenic cells. This one group, this is one, this is the next one, this is the next one. In between that, you can see a tall cell with a oval nucleus. No, that is the Sertoli cell. Okay, this is the Sertoli cell. In between that only you can see the groups of spermatogenic cells. So from the basal layer, which cell will be there? Can you answer this? Which cell will be there? They are the spermatogonium. Spermatogonium. What is the next cell? So this is the spermatocytes. These are the spermatocytes, mostly primary spermatocytes. Then you can see this is the spermatids. Then this is the spermatozoa. So these are the spermatogenic cells you can see. So what is this one? I told no. What is this one? Yes, this is the Sertoli cells. This is the Sertoli cells. Okay, within the seminiferous tubules, you can see all these cells. Okay, you understand this now? So spermatogonium give rise to spermatocytes. So we have primary and secondary spermatocytes. Secondary spermatocyte, it will disappear because its half-life is very less. Okay, so soon you can see the spermatids only. Spermatids give rise to spermatozoa. So on either side of spermatogenic cells, you can see the Sertoli cells. These are the pillar cells, supporting cells. Okay. Then apart from the seminiferous tubules, in between the seminiferous tubules, you can see group of cells here. No, they are the interstitial cells of Leydig. Interstitial cells of Leydig. They are responsible for production of testosterone. Okay, these are the cells that produce the hormone, male hormone, testosterone. So this is the interstitial cells. Okay, now we'll see about the Sertoli cells. Sertoli cells, it's also named as sustentacular cells. Sertoli cells, the name for that is sustentacular cells. We also call it as supporting cell. Okay, supporting cell. So how it looks like? They start from the basal lamina, they reach up to the lumen. It's also like spermatogenic cells. Okay, it starts from basal lamina. It reaches the lumen. They are the tall cells. They are irregular because they have the lateral processes. You can see the extensions, no? They are named as lateral processes. They are tall cells, starts from basal lamina to the lumen. They are irregular because of the presence of lateral processes. Okay, because of the lateral processes, we have two compartments formed. Okay, see in between what you can see, these are the spermatogonium. Then we have the primary spermatocytes. This gives rise to secondary spermatocytes. Spermatids are there, no? Here you can see up to the lateral primary spermatocytes, spermatogonium, primary spermatocytes, we have the lateral process, no? So here, for example, laterally in this part also, one more sertoli cell will be there. That also will have lateral process. These two lateral process, they will join. Okay, so this lateral process, it divides the compartment into basal compartment and adluminal compartment. Because this spermatogonium and primary spermatocytes, they are considered as the germ cells. They are already produced in the human body, so they are not foreign to the human body. But from the primary spermatocytes, the secondary spermatocytes, spermatids, spermatozoa, they are produced. No, they are considered as foreign to the body, foreign particle to the body, like an antigen. So when the antigen is produced, what will happen? Antibodies are produced. So this antibodies will agglutinate the antigens, okay, because they are going to kill that one, no? So if it agglutinate means the spermatozoa, it lost the function. So this will lead on to infertility. So to avoid that, the Sertoli cells lateral processes, they join with the adherent Sertoli cells, they form the blood testis barrier. They divide this into two compartments. This basal compartment, they have the spermatogonium and primary spermatocytes. They belongs to the body. Okay. But the secondary spermatids and spermatozoa, they considered as foreign particles. So they will be lying in the adluminal compartment. Because it divides, this lateral process divides this newly formed cells from our immune system. Okay, so it separates from the immune system. Okay, you understand this? So what are the functions of Sertoli cells? So first it gives the support to the germ cells, developing germ cells. So physical and also nutritional support to the germ cells. The next one is it phagocytoses the residual cytoplasm. When the spermatogonia gives rise to primary, secondary spermatocytes, no, some of the cytoplasm is released. So they will be phagocytosed by this Sertoli cells. That's also a function of Sertoli cells. Then it's the most important function. It establishes blood testis barrier. It separates the newly formed spermatozoa from the immune system. Okay, so it establishes blood testis barrier. And it secretes the fructose-rich fluid. It will nourish the developing sperms. It also secretes anti-Mullerian hormone. So anti-Mullerian hormone, what it do? It will inhibit the formation of the female reproductive system, because this Mullerian hormone is responsible for development of the female reproductive system organs. So it secretes anti-Mullerian hormone that will inhibit the formation of female reproductive organs. It secretes the inhibin. Inhibin is the one that inhibits the FSH. FSH is mainly important in female reproductive system for development of the follicles, everything. It produces hormones. So that is not needed in the male reproductive system. No? For that, the Sertoli cell, it secretes inhibin. That inhibin will inhibits the FSH. So these are the functions of Sertoli cells, supporting cell or sustentative cells. Okay, now you understand now? Yeah. So just a note on blood testis barrier. See here, this is the testis. Okay, testis. In the interstitial tissue, some capillaries will be there. They will be having the blood. Okay, blood cells, everything will be there. Okay, here you can see the base of the seminiferous tubule. You can see the spermatogonia, spermatocytes, secondary spermatocytes, then spermatozoa cells are there. This thing, see the pillar cells, this is the Sertoli cell. So the Sertoli cells, they divide this into two compartments, basal and adluminal compartment. So this newly formed sperms, they are not exposed to the immune system. So they are separating the blood system that will contain the immune cells. No? So it separates this system from the newly formed cells. So what happens if it is exposed means this is considered as foreign body. So within this vascular system, the cells will produce an antibody. So it will agglutinate, that will result in infertility. To avoid that, the Sertoli cells forms a barrier. Okay, so this when the sperms produce this immune system, it won't be sensitized, it won't be produced the antibodies. Now you understand now what is blood testis barrier? Yeah. Good. So this is the blood testis barrier. So what we have discussed now, we discussed about the seminiferous tubules. Oh, what are the spermatogenic cells? What are the Sertoli cells? We discussed. So apart from seminiferous tubules, what is there within the testis? That you can see the interstitial cells. So in this interstitial cells, they are also called as interstitial cells of leading cells. This is the seminiferous tubule. Here you can see the interstitial cells. So what are the functions of interstitial cells? What are the features of interstitial cells? They are large round polyhedral cells. They are, see you can see, they're a little bit big, no? They are large round polyhedral cells. And they have eccentrically placed nucleus. Here you can see this is eccentrically placed at one corner. Okay, so they have the eccentrically placed nucleus. They are lying in the connective tissue between the coils of seminiferous tubules. This is one coil of seminiferous tubule. This is one. In between the coils, you can see a connective tissue that contains the interstitial cells of leading. Okay. So what is that function? What is the function of interstitial cell? This secretes the testosterone. Okay, it secretes the testosterone. Okay, you understand now? So what you have to know about the testis, you have to know about the interstitial cells of leading and also seminiferous tubule. Within the seminiferous tubule, you have to discuss the Sertoli cells, spermatogenic cells. Okay, yeah. So these pictures show you the seminiferous tubules. You can see so many spermatogenic cells. Okay, so these are the seminiferous tubule. In between interstitial cells will be there. So you can see a slide like this one. So this is also called as germinal epithelium. So because these are the germ cells, no? So and it's also stratified because multiple layers are there. So it's a stratified epithelium, like okay. So these are the what you are seeing. Everything is the seminiferous tubule. This is the seminiferous tubule. This is the seminiferous tubule. This is also seminiferous tubule. Okay, so when we expand this, you can see like this. So first layer we have spermatogonia, then spermatocytes. Okay, this gives rise to spermatids, then sperms are produced. So it will be like this. So these are the seminiferous tubules. So germinal epithelium. This is also called as germinal epithelium. So in between seminiferous tubules, what you can see? So interstitial cells of leading you can see. Okay, you understand now? So this is also the seminiferous tubules and interstitial cells. So this is the seminiferous tubule, this is the interstitial cells. So you can see clearly you can see the Sertoli cells. So pillar cells in between a group of spermatogenic cells are there, no? Now you can tell. So that is, what is that? What is that arrow mark? So that is the Sertoli cells. What is the cell near the base? What is that cell? That is spermatogonium. Then what is the next cell? That is the spermatocytes. Okay, spermatocytes. Mostly primary spermatocytes only we can see in sections. Okay, what is that? That is the spermatids. So what are these things? These are the spermatozoa. Good. So now you can understand. You will answer the questions now in testis. So now you know about the testis. Now, next one, so testis we finished. Okay, the rest of the things we have to see now. The next one is epididymis. So you know the gross features of epidermis, no? It is a comma-shaped structure. You can see comma-shaped structure. Okay, it is also a convoluted tubule, lies posterolateral to the testis. This is testis. Posterolateral, you can see the testis. So it has three parts. So this is called as head, this is the body, and this is the tail of the epidermis. The tail of the epidermis, it continues as the vasculature. Okay. So now you can see the same thing. So this is the epidermis. It lies posterolaterally. You know how the epidermis is formed. The efferent ductules, they join and form the epidermis. Okay. So you can see the head of the epidermis here. This is the head. Then this is the body of the epidermis. Body of the epidermis. Then here you can see the tail of the epidermis. This tail of the epidermis, it continues as the ductus deferens. Okay. So first we'll see about the head of the epidermis. So the head of the epidermis, the tubules are there, no? They will be lined by ciliated columnar epithelium. It should have cilia because this epidermis will be transporting the sperms, no? So there will be ciliated columnar epithelium. When you see the body and tail, that is nothing but the duct of the epidermis. The duct of the epidermis only form the body and tail. That will be lined by pseudostratified columnar epithelium. Pseudostratified columnar epithelium with a special feature, that is the stereocilia. Stereocilia. They are long processes, ciliary processes. They are non-motile. Okay. So what is the lining epithelium in the head? That is ciliated columnar epithelium. In body and tail, there is pseudostratified columnar epithelium with stereocilia. So you can see like this, pseudostratified. You can see the multiple nucleus, no? So actually this is single layer only. Because of the presence of the nucleus, it looks like multiple layers. So that is called a pseudostratified columnar epithelium. So this is the pseudostratified ciliated columnar epithelium with stereocilia. Here you can see some projections of these are the stereocilia, hair-like processes. So here also you can see. So this is the pseudostratified epithelium. You can see small, minute stereocilia. Okay. Now it is, you know, the features of pseudostratified epithelium. Pseudostratified epithelium is there you can see a long cell and a short cell. Here, the long cells with the oval nucleus, they are called as principal cells. Principal cells. The short cells, they are called as basal cells. Okay, those principal cells only will be having the stereocilia. These basal cells, they act as stem cells. Okay, so from the stem cells only the rest of the cells are produced. Okay, then these ducts of the epidermis, they will be having smooth muscle layers. Okay, smooth muscle cells will be present here. Okay, then this is the stereocilia you already know. So these are the features you can see in each duct cell. Okay. So principal cells, you know the feature, it is the tall cell with basal nucleus. The principal cells only near to the lumen. So it will be having irregular microvilli and also non-motile stereocilia. Okay. So these are the tall cells. So this only will reach the lumen. There it contains stereocilia and irregular microvilli will be there. When you see the basal cell, they are short cells and they won't reach the lumen. See, these are the short cells. They will be present here only. They won't reach the lumen. So they are also called as stem cells. Okay. So these two cells, they will be lying over the smooth muscle layer. Okay. When you see the smooth muscle layer, the thickness will be increasing from head to tail because it has to propel the sperms. No, for that the thickness will increase. The smooth muscle layer, they are arranged as inner circular, outer longitudinal layers. So what are the functions of epidermis? The function is it phagocytoses the defective spermatozoa. So seminiferous tubules, they produce the sperms, they pass through the efferent ductules, then it reaches the epidermis. There, if some spermatozoas, they are defective, means that's not needed. So those defective spermatozoas, they are phagocytosed by the epidermis. That's the first function. Then if there is excess fluid is there, means it will absorb the excess fluid also. And main thing is the sperm matures in the epididymis. So maturation of sperms, they are happening in the epididymis. Okay, so these are the functions of epididymis. Okay, you understand now? Okay, now we are seeing the slide like this. Okay, so what you are seeing means, see, it also sometimes confused with the testis. Okay, in testis there will be germinal epithelium will be there, interstitial cells will be there. Okay, seminiferous tubules will be there. You sometimes you will be mistaken it as seminiferous tubules, but the thing is, in testis, in seminiferous tubules, sperms are produced. So you can see the sperms near this part. Okay, and you can see the Sertoli cells clearly, and spermatogenic cells, group of cells will be seen in between. But see here you can see the outer smooth muscle layer. Then this is the pseudo-stratified epithelium. It looks like a multiple layer, but it is not like that. It is single layer only. This is pseudo-stratified epithelium. And here only sperms are transported, no? So the maturation are taking place, no? So inside the lumen, you can see the collection of sperms in all lumen. So sperms are collected here. So inside, in the middle of the ducts, you can see the collection of sperms. So you have to differentiate the testis from the epididymis. So what is these rounds? They are the ductules of epididymis. Ductules of epididymis. So when you see each ductule, it is lined by what is that? Smooth muscle layer. Then what you can see, what is this one? What is that epithelium? Pseudo-stratified epithelium, seriated columnar epithelium. Then, so what is this one? What is this one? What is this collection? What is this one? That is the sperms. Okay. So when you see a slide, it will be looking like this. See, this is the pseudo-stratified epithelium. Okay, lining the duct of epididymis. Within this, you can see the collection of sperms. See, all the ducts, they are having the sperms. Okay, so this is the slide of epididymis. Okay, you understand now? So this is the slide of epididymis. So this is also an example showing the pseudo-stratified epithelium. This is the pseudo-stratified epithelium. Then you can see the collection of sperms. You can see clearly, no, sperms are collected here. Okay, so in seminiferous tubules, in testis, it's not like this, no. You can't able to see like this, no. Sperms are near the germinal epithelium, but here, see, the lumen is filled with sperms. Okay, so here you can easily see this pseudo-stratified epithelium. Inside that, you can see the sperms collected here. Okay, this is also a slide showing the pseudo-stratified epithelium. Inside, you can see the collection of sperms. Okay, so that's all about the epididymis. So two things we discussed. One is testis, it's over. The next one, what we discussed? Yeah, epididymis. So two things over. So what are the remaining things? What we have to discuss now? So this is over, no? These two things over. Now we have to discuss this long tube-like structure, that is the vas deferens. Then this one, this sac-like structure, that is the seminiferous tubule we have to discuss. Then this gland, accessory male reproductive gland, that is the prostate gland, that also we have to discuss. Okay, so vas deferens, first we'll see. Vas deferens is also called as ductus deferens. It's also called as ductus deferens. Okay, vas deferens also called as ductus deferens. So this vas deferens, it is a thick cord-like muscular tube. It's a thick cord-like muscular tube. See, from duct of epididymis, it starts in the scrotum. It passes through the, it passes along with the spermatic cord contents. So it passes through inguinal canal, then it reaches the peritoneal cavity, then it opens it joins with the seminiferous tubules, then it forms the ejaculatory duct, then it opens into prostatic part of the urethra. When you see the length of the vas deferens will be around 45 centimeter long. Okay, 45 centimeter long. So from where it starts from the lower end of epididymis and it opens into prostatic part of the urethra. Okay. So now we'll see the histological feature of vas deferens. So when you cut the tube, it's a tube-like structure, no? So when you cut a tube, you can see a lumen. Okay, so you can see a lumen. So when you see it, it will be like this. A lumen will be there. Then you can see some structures. Okay, so when we make a section and strain the slide, we can able to see 3 layers. The first one is mucous membrane near the lumen. The next one is muscle layer. So this is the muscle layer. The third one is adventitia. The outer one is the adventitia. These 3 things you can see now. So first layer is mucous membrane. The next one is muscle layer. The third layer is connective tissue layer. Okay. So now we are going to see this. See what is the shape of the lumen? How it looks like? It somehow looks like a star, no? So it will have a star-shaped lumen. And also, mostly this should be the muscle layer, no? See, thick muscle layer is there. Why it has to be thick? Because it's carrying the sperms against gravity, no? So it should have strong musculature. So the muscle fiber layers should be more. Okay, so with this idea, we'll discuss this 3 layers. So that is the mucous membrane. The next one is muscle layer. The next one is adventitia or connective tissue layer. These 3 things we are going to see. So first one is mucous membrane. So you know star-shaped, irregular star-shaped lumen will be there. So you can see we are focusing a small part of epithelium only. So here you can see irregular star-shaped lumen. Then what is the lining epithelium here? This is pseudo-stratified columnar epithelium with stereocilia. In epididymis also you can see the stereocilia. In vas deferens also the lining epithelium will be same because it's a continuation only. So pseudo-stratified columnar epithelium with stereocilia you can see. So this is the epithelium. Beneath that epithelium you can see the lamina propria. Okay, you understand this? Now we are discussing about the vas deferens. In the vas deferens, 3 layers are the mucous membrane, muscle layer, then connective tissue layer. In mucous membrane, as you know, mucous membrane is made up of epithelium and also lamina propria. Here, epithelium is pseudo-stratified columnar epithelium, and a special feature will be that is the stereocilia. Beneath that, you can see the lamina propria. This lamina propria is made up of collagen fibers and elastic fibers. Collagen fibers and elastic fibers. Okay, is it clear for you? We discussed now about the mucous membrane. The next one, muscle layer. When you see the muscle layer, it's made up of smooth muscles. Okay, it is made up of smooth muscles. They are arranged as inner longitudinal, middle circular, outer longitudinal. So this I already told you, the smooth muscle layer, it is very thick because it has to act against the gravity. So it is arranged in 3 layers. The inner and outer, they are longitudinal fibers. Middle layer is circular fiber. When you see the adventitia, it is made up of loose areolar tissue. Within that loose areolar tissue, some blood vessels, nerves will be there. So you already know the function of the vas deferens. It propels the sperms during ejaculation. So this is the picture showing the section of the vas deferens. You can see the irregular star-shaped lumen. So this is the irregular star-shaped lumen. So you can see the lining epithelium. Okay, this is the lining epithelium. So that is pseudo-stratified columnar epithelium with stereocilia. Beneath the lining epithelium, what will be the lamina propria? It is made up of collagen fibers and elastic fibers. What is the next layer? That is the muscle layer. How many layers are there? Yeah. 3 layers are there, no? They are inner longitudinal, outer longitudinal, middle circular layer. Okay, so this muscle fiber layer is very thick. So inner longitudinal, middle circular, outer longitudinal. What is the next layer? That is the adventitia. That is the connective tissue layer. It's made up of loose connective tissue. It contains some blood vessels, some nerves. So this is the connective tissue layer. See, when you see a section of ureter and vas deferens, you may confused. The thing is, in vas deferens, this muscle layer will be very thick. Okay, it will be very thick. When you see the lumen, a little bit, both are a little bit star-shaped only. But what is the thing you have to note here? In vas deferens, we can able to see the pseudostratified epithelium. In ureter, you can see the transitional epithelium. That difference is there, no? Okay, so during the examination, you have to concentrate. You have to see the muscle layer thickness, and also you have to look for the lining epithelium. Okay, is it clear? Vas deferens. So vas deferens is over. So we finished the vas deferens. So what are the things we discussed up to now? We discussed the testis. Epididymis we discussed. Vas deferens also over. The next one is prostate gland. This is the largest accessory sex gland. Okay, so where it is located? Near the neck of the bladder. Okay, you can see here, this is the prostate gland. Okay, within the prostate gland, you can see the prostatic part of the urethra. Okay, so what are the histological features of prostate gland? So this line you have to know, it is a fibromusculoglandular organ. Fibromusculoglandular organ. Because it is made up of fibrous tissue, muscular tissue, and also glandular tissue. So three things they combine and form the prostate gland. Okay, so this glandular part will be more. So this is this part of prostate gland. When we cut it coronally, and when you see it will be looking like this. So prostate gland will be covered by a capsule. Okay, true capsule and false capsule will be there. You can see false capsule, true capsule, some venous fluxes also there. This yellow color is the gland, this prostate gland. Within the prostate gland, you can see these things are the glands. Okay, so around 20 to 50 tubulo-alveolar glands are there. 20 to 50 tubulo-alveolar glands are there. You know already about what is the tubular glands, alveolar glands. Okay, tubular means they will be like a tube. Alveolar means they will be cone shaped. Okay, so both the things will be there. And how it is arranged? This middle part, this is the prostatic part of the urethra. The zone that is near to the prostatic urethra, it contains mucosal glands. They will have the duct. That duct directly opens into the prostatic urethra. Just see here, mucosal glands. In the next layer, you can see the submucosal glands. They drain into a duct. That duct will be draining into the prostatic urethra. The peripheral zone is the prostate gland. That is the prostatic gland proper. They will have a long duct. That duct will finally open into the prostatic urethra. So within the prostate gland, we can able to see 20 to 50 tubulo-alveolar glands. This tubulo-alveolar glands, they are arranged in three zones. One is named as mucosal glands. They directly drains into the prostatic urethra. This submucosal glands and prostatic gland proper, they drain into a duct, long duct. That long duct, it finally opens into the prostatic urethra. Okay, you understand now? So when you see the glands, each gland will be lined by simple columnar epithelium. Simple columnar epithelium. Okay. So you know it's a fibromusculoglandular organ. First, glandular part we discussed now. We have to discuss the remaining part, fibromuscular. What it is made up of? It is made up of smooth muscle is there, collagen fibers will be there, and also elastic fibers. So three things will be there. smooth muscle, collagen fibers, elastic fibers. When you see, they are not uniformly arranged. They are arranged in different directions. Okay, in different directions they will be arranged. And these fibromuscular stroma will contain blood vessels, lymph vessels, and also nerves. Okay, so this is about the fibromuscular stroma. When you see the gland, in few glands you can see some collections. Okay, that is nothing but the prostatic concretions. These glands will produce some secretions. No, in elderly male, these secretions they condensed and they form the hard part that is named as prostatic concretions. It is also named as corpora amylaceae. Corpora amylaceae. Okay, corpora amylaceae. So, in prostate gland, what are the things you have to discuss? This is a fibromuscular glandular organ. So, fibromuscular part, it is made up of smooth muscle, collagen fibers, elastic fibers. They are arranged in different directions. They contain blood vessels, lymph vessels, nerves. When you see the glandular part, they are containing 20 to 50 tubuloalveolar glands. These glands are arranged as mucosal glands, submucosal glands, then prostatic gland proper. Within the glands, they are lined by simple columnar epithelium. Within that, few prostatic concretions will be there. They are named as corpora amylaceae. So this is a slide of prostate gland showing the fibromuscular stroma. These are the glandular part. This is the prostatic concretions. That is the corpora amylaceae. Okay. So this is the prostatic secretion. This one only in elderly age, this becomes the prostatic concretion. So everything, they are the glands. This is the fibromuscular stroma. Okay. So what are the functions of this prostate gland? Means it will produce thin fluid. That thin fluid is made up of acid phosphatase, amylase, fibrinolysin, prostate-specific antigen. Okay. This fibrinolysin, what is the function of it? Means it liquefies the sperms. Okay, so the sperms will be along with some mixture of fluids, no? So it liquefies. Okay, the prostatic secretions, they facilitated by the contraction of smooth muscle stroma at the time of ejaculation. So prostatic secretions, they will be passing along with the ejaculation, along with the sperms. This prostatic secretions, it promotes the mobility of the sperms also. These are the functions of the prostate gland. So you can see in this section, you can see the prostatic urethra. This is the prostatic urethra. This is the utricle, okay, ejaculatory duct you can see. So the rest of the things you can see the fibromuscular stroma. You can see the glands, prostatic glands. Okay, you understand now? So this is also a slide showing the prostate gland. So these empty spaces are nothing but the glands, prostatic. Within the prostate glands, you can see the glandular part like by simple columnar epithelium. So they are the glandular acini. This is the fibromuscular stroma. Okay, so this is also a slide of prostate gland showing the glands. So everything, these are the prostate, this glandular part. This is the fibromuscular stroma in between. Okay, so prostate gland is over now. The next slide, this is the last thing we are going to discuss now. That is the seminal vesicle. Okay, seminal vesicle. So this is the elongated sac-like structure. This is the elongated sac-like structure. When you cut open, you can see highly convoluted irregular lumen. See, when we cut the seminal vesicle, inside that, it will be like this. It is highly convoluted and the lumen will be very, very irregular. Okay, it's very irregular. So this is also made up of 3 layers: mucosa, muscle layer, and adventitia. Okay, so when you see the mucosa, so I don't know that is very irregular, no? So this mucosa is thrown into highly complex folds. It is thrown into highly complex folds. Those folds are named as crypts. Okay, so it will be lined by pseudostratified columnar epithelium. So epidermis lining epithelium also pseudostratified, vas deferens also pseudostratified. In seminal vesicle also, the lining epithelium is pseudostratified columnar epithelium. So the feature is highly complex folds with which forms the crypts. Lining epithelium is pseudostratified columnar epithelium. When you see the muscle layer, It is arranged as inner circular or outer longitudinal smooth muscle layer. Inner circular, outer longitudinal smooth muscle layer. Adventitia, it is made up of loose connective tissue. So these three layers only you can see. So this is the picture of seminal vesicle. See, this is the mucosa part. This is the muscle part. This is the adventitia part. When you see the mucosa, how it will be look like? It is thrown into numerous highly complex folds. So these are the folds. So usually we divide this into primary mucosal folds and secondary mucosal folds. So these folds will be lined by pseudostratified columnar epithelium. Okay, then lamina propria will be there. So these folds, in between the folds, there are some gaps. No, this is named as crypts, mucosal crypts. Okay, these are the folds, primary and secondary folds. These folds, it forms the crypts. Then we have the muscle layer, that is inner circular and outer longitudinal muscle layer. The last one is the adventitia layer. It will contain some loose connective tissue and blood vessels will be there. You can see here. So this is the picture of seminal vesicle. So this slide also showing the seminal vesicle. This is also seminal vesicle. You can see the lumen and you can see irregular complex folds. So here also you can see irregular complex folds. Okay. So this is also a slide of seminal vesicle. This is a sac-like structure. When you see, you can see the irregular complex primary and secondary folds. Okay. So now we discuss the histological features of testis, epididymis, vas deferens, prostate, seminal vesicle. Everything is so. Now, few things we'll know about the functional histology. So in testis, I have already discussed what is blood testis barrier. Okay, so we have to discuss about the sperm specific antibodies. So you know the secondary spermatocytes, spermatids, spermatozoa. These three things, they are considered as the foreign particles because it has a specific proteins. It possesses some specific proteins. Okay, this proteins, it is considered as foreign to the body. If something considered foreign to the body means what our immune system will do, it wants to fight against that. So it will produce the antibodies. So if antibodies produced for the sperms means what will happen? The sperm will agglutinate. If sperm agglutinate means it won't function. It will result in infertility. So that is very important. So secondary spermatocytes, spermatids, spermatozoa, they contain specific proteins. It is isolated from the testis barrier by the Sertoli cells. If it breaks, means what happened? Formation of sperm specific antibodies. This will agglutinate sperms and it will result in infertility. Okay, this is the one we have to know. The next one is syndrome is there, that is named as immotile cilia syndrome. Immotile cilia syndrome. In this case, the sperms will be immotile, so it won't function. If the sperms are not immotile, means it won't enter in the uterus and all, no? So it will result in infertility. What is the reason for that? Means there is lack of dynein protein in the sperms. Okay, this dynein protein is required for the motility of cilia and flagella of the sperms. If it is not present, means the sperms are immotile. This immotile cilia syndrome, it will have some other abnormalities like bronchiectasis. Bronchiectasis means dilatation of bronchus. So these are group of clinical conditions they are seen together. Okay, so this is the one. This is called as immotile cilia syndrome. The next one is benign prostatic hypertrophy. So now you know prostate is made up of glandular tissue and also fibromuscular stroma. If the glandular tissue proliferate, means in elderly people, what happens? It will compress the prostatic part of the urethra. No? So the patient will have incontinence. They will feel difficulty in passing urine. Okay, so the benign prostatic hypertrophy, it occurs due to proliferation of glandular tissue of the prostate. It is mostly seen in the elderly people. Okay, so they will come with complaints of difficulty in passing urine. Okay, the next clinical condition is adenocarcinoma of prostate. You know, prostate gland, it has glandular tissue. You know, so when carcinoma, it develops in a glandular tissue, means that is named as adenocarcinoma. So this prostate gland, it's a gland, no? So in glandular tissue, some tumors may arise. If it arises, means that is named as adenocarcinoma of prostate. If you investigate, if you do some investigations, mean blood investigation, means in that person you can see the increased prostate specific antigen. If there is increased prostate specific antigen, means it will be diagnosed as a case of adenocarcinoma of the prostate. These are some functional histology you can add up along with the histological features. Okay, is it clear for you? So the next one is unpresented testis. This is the final one. So usually testis, it lies in the scrotum. Why it lies here? If it is in abdominal cavity, means in abdominal cavity the pressure, the temperature is more. So the sperms, it won't tolerate the temperature. So it has to away from the temperature. That's why it is in the scrotum. So if the testis is not in development, in embryology itself, we discussed about how the testis descends, no? So you know that. So if the testis, it is not present in the scrotum, it is still in the abdomen, means the abdominal temperature will inhibit the germ cell production. So this germ cells, it won't produce. So this will leads on to male infertility. So that's a case called as unpresented testis. So these are some functional histology you have to know. Now can you identify this slide? So everything we discussed, now you have to identify the slide. What is this slide? What you are seeing here, these are the semiferous tubules. Okay, you can able to see the germinal epithelium. Okay, here a few sperms are there. Okay, in between you can see the interstitial cells also. So what is this slide? This is a slide of testis. Good. The next slide, you can see some ductules. Okay, within that you can see collection of sperms. Okay, sorry, collection of sperms you can see here, no? So and also see the lining epithelium, pseudostratific, the multiple layer, no? So this is pseudostratific. So what is that slide? This is a slide of, yeah, say epidermis. Now you can see a luminal structure. You can see as irregular star-shaped lumen, pseudostratified epithelium is there. You can see thick muscle fiber in the longitudinal, outer longitudinal, middle circle. What is this slide? This is a slide of, yeah, it's a vas difference. What is this slide? You can see here some glandular parts are there, some fibromuscular stroma is there. So this is the prostate gland. Okay. Then here you can see highly complex folds. Okay, highly complex folds you can see here. So what is the slide? This is a slide of seminal vesicle. Good. So you understand this structures, no? So histology of male reproductive organs. Okay, any doubts here? Okay, no? So if there is any no doubts, means we'll conclude this